Hello, everyone. I want to introduce Dr. Andrew Feigen. He is a professor of neurology at NYU Langone Health and director of the Marlene and Paolo Fresco Institute for Parkinson's and Movement Disorders. He also serves as the chair of the Huntington Study Group. Thank you for being with us today, Dr. Feigen.
Thank you very much. Thank you to all of you for joining and coming to listen to me today. And thank you to HD Reach for inviting me to speak. So I'm really speaking on behalf of, as a representative of the Huntington Study Group today. And I want to start by telling you a little bit about the Huntington Study Group, but I'm really going to spend the majority of my time today telling you about our two active clinical trials that we're recruiting and looking for patients to participate in. So let me go ahead and start just telling you a little bit about the Huntington Study Group. The Huntington Study Group was formed in 1993 by Ira Schulson and Jack Penny. And the idea of forming the network, the clinical trial network of the Huntington Study Group in 1993 was the recognition that with the advent of potential new therapies for Huntington's disease, we were going to need to be able to do multi-center clinical trials. We were going to have people working together from all over the world, really, to collaborate on doing these big multi-center clinical trials. And so the network was formed called the Huntington Study Group. Over the last 28 years, we've grown a lot, and we now have over 800 members. Not everybody who are members of the Huntington Study Group are investigators or coordinators in clinical trials. We also have a lot of scientists and other kinds of experts and caregivers who are members of the Huntington Study Group as well. And we all are working together to try to expedite the study of and hopefully delivery of new therapies for Huntington's disease. We have 130 credentialed Huntington Study Group clinical trial sites all around the world. And we have conducted 38 either observational studies or clinical trials, including the only two trials that have led to FDA-approved drugs for Huntington's disease. So I could actually spend an hour telling you about what the Huntington Study Group does, but today I really wanted to focus on the clinical trials that we have, our active clinical trials. So first I'll just show you the sites in North America. We have 106 study sites in North America, in the US and Canada, and this is where they're located. So I'm going to start by telling you about one of our active clinical trials, the CONNECT-HD trial. The CONNECT-HD trial is a double-blind, randomized, placebo-controlled clinical trial that is aiming to study the effectiveness of a drug called valbenazine for chorea associated with Huntington's disease. And of course, we'll also be looking at the safety and tolerability of valbenazine in individuals with Huntington's disease as well. So what is a randomized, double-blind, placebo-controlled clinical trial? Well, I'll take the first word first, randomized. Randomized means that people who participate in the trial are randomly assigned to either get the study drug or get a matching placebo, something that is really kind of a fake medication, but it looks and is indistinguishable from the active medication. The reason the randomization is important in these trials is that if people are randomly assigned to one group or the other, if there are enough people enrolled, those groups will be matched for disease severity and chorea severity and for age and for gender and really for everything you can think of. If you randomly assign people to different groups, those groups will be matched hopefully in every important way. And so you'll be looking at matched groups and their response to the drug versus placebo. Double blind means that neither the patients who participate in the trial nor the doctors who are evaluating the patients know who's on the drug and who's on the placebo. And this is done to remove bias in the trial so that when we're assessing people during the course of the trial, we're doing it without any biases about whether we think the drug might be working or not working. And it's to keep us honest in terms of determining whether the drug is working. So those are the key elements to these big efficacy double-blind trials. So what is valbenazine? Valbenazine is actually an FDA-approved drug. It was approved in 2017 for the treatment of a condition called tardive dyskinesia under the trade name Ingrezza. It's a prescription medication used to treat adults with involuntary movements in the face, tongue, or other body parts that cannot be controlled, which is called tardive dyskinesia, usually a side effect of a class of medications. And the tardive dyskinesia actually can look very much like the chorea of Huntington's disease, and there are good reasons to think that this drug, which is been proven to be helpful for tardive dyskinesia, could also be helpful for the chorea of Huntington's disease. And it's now being studied for the treatment of chorea associated with Huntington's disease. But it's important to point out that valbenazine to treat is not FDA approved for the treatment of chorea of Huntington's disease. The point of this trial is to try to get to the point where it could be approved for the treatment of chorea of Huntington's disease. Potential side effects of valbenazine are mainly related to drowsiness, tiredness, and sedation. The trial will enroll approximately 120 patients who meet certain criteria. We're including men and women, and you have to be between the ages of 18 and 75. You have to have a diagnosis of Huntington's disease, and you have to have troublesome or bothersome chorea. That there's specific definitions of that in the trial, what are called inclusion and exclusion criteria in the trial. And of course, you have to be willing and able to comply with the study instructions and procedures. The study will include 9 visits that can last up to 18 weeks. That includes 4 weeks of a screening procedure. Screening procedures in clinical trials are used to make sure people don't have any active medical problems that might interfere with the conduct of the trial, make sure people are eligible for the trial. So there's a period of 4 weeks in which there's a visit where you get screening tests done, and then you'll be scheduled for the baseline visit. There'll be about 12 weeks of study drug dosing, and then 2 weeks after that, after this last dose of the study drug is taken, there will be another visit at the end of 2 weeks after that. So that comes to a total of 18 weeks. The study will be provided to you as part of this if you participate in the trial, and you'll take it at home on your own once a day. The study is taking place at 46 Huntington Study Group sites throughout North America and US and Canada. If you are interested in participating, you know somebody who might be interested in participating, you can find out the nearest site to where you are by either going to www.connecthd.org or hdtrialfinder.org or clinicaltrials.gov. Or you can call the Huntington Study Group at a toll-free number listed here, or email us at info@hsglimited.org. I'm now going to shift gears to another big trial that we're doing at the Huntington Study Group called PROOF-HD. PROOF-HD stands for Predopidine Outcome on Function in Huntington's Disease. This is a phase 3, meaning it's a trial that's trying to prove that the drug works. Randomized, double-blind, like we talked about earlier, placebo-controlled. Parallel arm means that there's 2 groups. One group will get 45 milligrams twice a day of predopidine, and the other group will get placebo, and they'll be followed in the same way during the course of this study. It's a multi-center study taking place at 60 sites, 30 in Europe and 30 in North America. And it's going to be evaluating the efficacy and safety of this drug called predopidine in patients with early stages of Huntington's disease. So this is a complicated slide. I'm not going to go through it in detail. Just the main point is that the primary mechanism of action of predopidine is that it works at a receptor called the sigma-1 receptor here that says S1R. That's something called the sigma-1 receptor. And the sigma-1 receptor is involved in a lot of pathways. If you can see on the left panel here, this is kind of, I'm not going to go into the technicalities of these things, but these are mechanisms that we think are helpful for preventing neuronal or brain cell death. And these are multiple mechanisms, and the sigma-1 receptor seems to be involved in that. And drugs that act at the sigma-1 receptor, we're hoping, will increase the ability to protect brain cells in Huntington's disease. Sigma-1 receptors are distributed throughout the brain. You can see this on the right, but especially in regions that are affected in Huntington's disease, in a region called the striatum, which consists of the caudate and the putamen, are especially rich in sigma-1 receptors. This study showed that on the left panel, with increasing doses or concentrations of the study drug, predopidine, this is in transgenic a mouse model of Huntington's disease. These are mice that have the Huntington gene expansion, and in orange, and the blue is mice that don't have the Huntington gene expansion. And so if you look at on the left side where it says 0, so the mice did not receive any of the predopidine, a lot of the brain cells are dying in those mice, up to 35 or 40%, as opposed to animals that don't have the Huntington gene mutation. Something like 15% of their neurons die. But as you increase predopidine doses on the left panel, as you move across to the right, where you go from 0.001, 0.01, 0.1, 0.51, a number of brain cells that seem to die starts to approach what happens in animals that don't have the mutant Huntington. So increasing doses of the drug seems to protect brain cells from dying. And on the right side, it shows that if you take the same animals, but you knock out the sigma-1 receptor, meaning these animals still have the Huntington mutation in orange here, but now they don't have the sigma-1 receptor, predopidine doesn't seem to work in the same way. So you need this. It does seem that the sigma-1 receptor is key for the effect of predopidine to work in terms of protecting the brain cells or neurons. Predopidine has been studied extensively in Huntington's disease, and has been given to many, many subjects. More than 1,300 patients in the past have received predopidine for, in many cases, a year or longer. And most of these patients had Huntington's disease. The doses ranged from 10 milligrams twice a day to 112.5 milligrams twice a day. And it's been found to be very safe and tolerable. And the dose we're using in the PROOF-HD trial of 45 milligrams twice a day actually has been found to have what we call placebo-like, AE stands for adverse events, like side effects profile. The side effects for the predopidine at 45 milligrams twice a day is really not different from a fake medication, from placebo. And this is based on quite a lot of experience. I'm going to turn to the primary outcome measure of the PROOF-HD trial, which is total functional capacity. This is a measure that's been around for a long time and used in clinical trials for Huntington's disease for about 40 years. We call it TFC for short. It's part of the Unified Huntington's Disease Rating Scale. This slide just basically shows you that the FDA believes that the TFC is a very important outcome measure for clinical trials, and that if a drug can be shown to be helpful for TFC, it's likely that will be meaningful to patients. The TFC changes in early Huntington's disease. The TFC changes about 1 point per year, declines by about 1 point per year in Huntington's disease stage 1 and stage 2. As people have more advanced Huntington's disease, the scale changes more slowly. So in this trial, we've decided to include people with stage 1 and stage 2 because we'll be more likely to be able to see an effect of the drug, since people will be declining at more of a rate of about 1 point per year. This shows you some data from an earlier trial that shows that people who, in an earlier trial called PRIDE, the people who got placebo, just as expected, declined at around 1 point per year. But the people who received 45 milligrams twice a day of predopidine over the course of that year did not progress. And this was highly statistically significant. This, unfortunately, was a secondary outcome measure of this trial, so it can't be used as conclusive proof that predopidine works. But this is really the basis for the PROOF-HD trial, to really try to nail down and prove with the TFC as the primary outcome measure that this drug may slow the progression of functional decline in Huntington's disease. This shows that in a prior trial, again, this is a kind of a post hoc secondary type of analysis, but a prior trial of predopidine 45 milligrams twice a day had an open label extension called Open Heart. So people took the drug for up to 5 years, and over 5 years, their total functional capacity declined by just under 2 points, whereas Other trials have found that people not treated with pridopatine decline as in the way we'd expect by about a point a year, about 5 points over 5 years. Again, suggesting that maybe being on this drug could be good over the long haul for patients with Huntington's disease. So what is PROOF-HD? I mentioned already, it's a phase 3 parallel arm randomized double-blind placebo-controlled multi-center study to evaluate the efficacy and safety of pridopatine in patients with early stage Huntington's disease. There are 2 arms, 45 milligrams twice a day of pridopatine and placebo. The target population is people with Huntington's disease who have total functional capacity scores of greater than or equal to 7. The primary endpoint is the change from baseline to week 65, about a year and a quarter in total functional capacity. But we'll also be looking at the percentage of people who don't change at all over that course of period of time. And there are a bunch of other assessments that we'll also be doing. So I'll just say that it's a partnership. The study's being done by the Huntington Study Group and a group of 60 investigators and coordinators from around the world, really, Europe and North America. I'm the PI for North America. Dr. Ralph Reilman is the PI for Europe. Dr. Sandra Kostic is the co-PI for North America, and Dr. Ann Rosser is the co-PI for Europe. I just want to put on the last slide in case people are interested. This is who you should contact, the contact information if you're interested in participating. Thank you so much for your attention.
Thank you so much, Dr. Fagan. We really appreciate all the great information and knowing about all the great trials that are going on right now. This is really, there's so much good work going on. I appreciate your bringing this to us.
Okay, well, thank you so much for having me. Appreciate it. Yeah, thanks.
Thanks.
Take care. Bye.